Hi class, and welcome to week 10 and lab 10. This is a recorded lecture. I wasn't able to meet this week because I had a doctor's appointment. So in this week, we're going over the immune system and infectious disease. So you'll be looking at the different structures of the human immune system, describing their functions. We'll look a little bit about epidemiology and public health. We'll look at a real-world scenario related to infectious diseases. And then you'll demonstrate the ability to apply the process of scientific inquiry and experimental design. So here we have the immune and lymphatic system structures and functions, and you can use your course textbook as well as other resources to kind of help you as you're going over the immune system. We have the tonsils, the thymus, the spleen, bone marrow, lymph nodes, and lymph vessels, and basically your lymphatic system is kind of like your body's kind of cleanup system. So it helps you clean up anything that's being broken down in your body. It helps with white blood cell production. So we kind of put our lymphatic system and our immune system as one in the same. So what you need to go through is identifying the structures and then be sure to tell me where they're located as well as their function. And again, you might need to use your textbooks or Google or other resources to kind of fill out specifically where these organs in the immune system are located and then what their function is. Then you will label the structures of the immune system and the lymphatic system in the image below, and you can use pretty much the picture from the above. It's just kind of another way to test you to make sure that you understand what are the organs of the immune system and where they're located. And then we'll look at the skin a little bit. Remember, the skin is your body's largest organ, and it's also your body's first line of defense because it keeps a lot of harmful substances out of the skin. And you are going to observe two cultures. So we've created two Petri dishes here, recording the data in the table provided, and then recording any observations with respect to the size, color, texture of what you see in these Petri dishes. So basically, what you're looking at is you may have both bacteria and fungi present on either of these plates in the unwashed hands and the washed hands. And to differentiate bacteria from fungi, look at the colors and sizes of the colonies. Bacterial colonies tend to be smooth and shiny looking, whereas fungi tend to look fuzzy. So these little white dots are most likely bacterial colonies. And then if you see kind of a fuzzy kind of colony, that's more of a fungi colony. So we've created these two Petri dishes, and you can read how we've created these. They were inoculated by pressing a hand against the nutrient agar in the plate. The first plate was inoculated with unwashed fingers, and the second was inoculated after the hands had been washed with soap and dried using a paper towel. So pretty amazing difference here between the unwashed and the washed hands. Also, notice that on the right, washing your hands doesn't eliminate 100% of microbes. And it's also possible that there was a contamination of the culture medium, which would impact the results. So there are still some bacteria present if there was some sort of contamination. So then you go through and you have to identify the number of the kinds of bacteria and the number of kinds of fungi in the unwashed and the washed hands. And to do that, they're asking you to count the number of colonies. And again, you'll have many colonies in the bacteria, maybe because there was contamination, but obviously you still have many more fungi colonies on the left-hand side. Some of these fungi colonies you can't even differentiate from themselves because they're just kind of growing into each other. So if you notice, one fungi colony looks about this big, and that's taking up the whole plate. You can take 1, 2, 3. I mean, there's probably, 50 fungi colonies. You see a couple bacterial colonies as well. So you'll write your data down there, just counting the number of colonies of the fungi and the bacteria you see. And that is your data. And I'm trying to think if there's anything else. In here, you can describe where there might be some discrepancies. So maybe there was contamination. That's why we still have a lot of bacterial colonies in the washed hands, and any other observations that you noted between the Petri dishes. And then epidemiology, and let's take a look at our lab guide. So in the lab guide, again, I think pretty much it's self-explanatory, telling you to use your lecture notes. The worksheet really takes you through everything you need to know. Epidemiology, you'll read the introduction and then follow the procedures to answer the questions. And then there's an infectious disease simulation. So let's go back to the worksheet, because I think the worksheet gives you all the information you need. So epidemiology specifically is a branch of medical science that involves the study of disease transmission within populations. And this is very pertinent to us because of COVID, but when we identify individuals who are ill, we can track them and then track where the illnesses occur. And then public health officials are able to provide education and guidance to the general population in order to reduce the risk of infection, whether that's wearing a mask, washing your hands. There are a number of ways that disease may be transmitted, such as contact with a contaminated surface, with contaminated water or air, or direct person-to-person contact. And this activity will utilize 2 different online simulators to demonstrate infection through person-to-person contact. And as a result of recent events, you likely have heard the terms below, and I want you to just define these 2 terms before you begin the exercise. And you can search them online, and you can write, what is an epidemic, what is a pandemic, and what does flattening the curve mean? Then view the images below captured from this Twitter. And in terms of the number of cases and the capacity of the healthcare system, what happens when our response shifts from whatever, it's just a cold or flu, to don't panic, but be careful. And that's what we mean by when we say we flatten the curve. We're just trying to decrease the number of cases so that our healthcare system isn't reached to capacity. Then we'll look at the following website. You don't need to subscribe to the Washington Post to view this article. It's available in multiple languages, and questions should be answered in your own words, not copied and pasted from the article. So you'll read through this article in the Washington Post. Why are health experts concerned about the exponential curve that we've experienced in COVID cases? This was between January 22nd and March 13th of last year. What can we do to slow the spread? And then you can view the disease simulations on this same webpage. The first 2 show a population of 5 individuals. Continue reading the article until you get to the simulation showing the population of 200, and take a note of the slope of the curve for sick individuals. Continue reading until you get to the simulation showing the population of 200 that was put under forced quarantine. And according to the article, why didn't this stop the spread? Continue reading. When you get to social distancing is deployed, take note of the slope on the curve for sick individuals. And basically, you're just kind of answering questions based on the article and using those simulations in that article. At the end of the article, the author will state, like a ball bouncing across the screen, a single person's behavior can cause a ripple effect that touches faraway people. And what does he mean by this? And then the last part, we will do an online simulation using an infectious disease simulation showing the transmission of a fictitious disease. So this is not real, it's fictitious, influenza, which mimics the transmission patterns of influenza. And the overview background information can be shown here. You do not need to download the data table file provided on this page. We're only going to complete a portion of the simulation, and then you'll record your data in the table provided. So you'll go to the following website, which contains the simulator, and the link should work, and I'll check that. And it's showing right away. I'll share my screen. So the link should bring you to a page that looks like this, showing a pandemic, and you'll go through and you'll read through the instructions about how this pandemic will spread through the community, listing out the dead, the immune, the at-risk, and then the contagious. So it's really a neat simulation. Have fun with it, especially, as we talk about COVID and how that's spreading. So the instructions are very clear. So I would take your time, read through the instructions as you work through that. If you have questions, let me know. And then you'll record some data. So the number of contagious individuals per day 0 to 25 with high density and high mixing. So be sure to kind of record your data as you're working through the simulation, the number of contagious individuals per day with low density and low mixing. And then once you have collected your data, you'll create a line graph to show the number of contagious individuals per day. So the day number will be on the x-axis with the number of individuals on the y-axis, and use one color for the line showing high density and high mixing, and a different color to show low density and low mixing. So your graph title would be labeling something like contagious individuals per day. And on the x-axis, which is on the bottom, you'll put 0 to 25 days. And then on the y-axis, which goes vertically, you'll put the number of contagious individuals. So you'll have kind of 2 points for each day, because you're doing 0 to 25 with low density and low mixing. So that'll be 1 point for day 0, for day 1, for day 2. And then with high density and high mixing, you'll have the second point. So you'll kind of have 2 points listed for each day, and then you'll connect the points as a line graph, and you can use a different color for each line. And then you have a couple of questions to answer at the end. So this is a really interesting lab, especially with what we've been through over the past 2 years. So have fun with it. Let me know if you guys have any questions. Like usual, this will be due on Sunday, as well as the quiz from last week. Keep up the great work, guys, and we will see you next week in person. And let me know if you guys have any questions.